Hi guys, this is the BioKid here with another video. This time we talk about the heat shock proteins and heat stroke necrosis. So let's get started. What are heat shock proteins? Heat shock proteins are stress proteins that are expressed by a cell in stressful conditions like heat, heavy metal stress, or hypoxia. These are highly conserved proteins and are found in all organisms. Some heat shock proteins also helps in protein folding for their proper functioning, and thus they act as chaperones. Heat shock proteins are classified based on their molecular weight, and some of them are HSP 60, HSP 90, etc. These play a key role in human diseases such as aging and diabetes. Now, what is heat stroke? Heat stroke is a life threatening condition that is characterized by the collapse of thermoregulation in the body. It mostly occurs due to hyperthermia. Heat stroke kills by necrosis, by disrupting the cell of its proper biological function. Hence, multiple organ failure takes place. An experiment is carried out on Caenorhabditis elegans, a model organism that is, and it is also a nematode, a batch of which was exposed to heat shock for 40 degrees centigrade for 15 minutes. What this experiment did was that kill almost all the population, leaving only 25% of them. Interestingly, the cell showed markers of necrosis. Preconditioning defense against hyperthermia. Now, what the scientists did was that gave them mild treatment before giving them the heat shock. The mild treatment was done at 34 degrees centigrade for 30 minutes. What this preconditioning did is that it activates certain transcription factors called the heat shock factor. More specifically speaking, it's heat shock factor 1 that aids ultimately the production of heat shock proteins. This creates resistance in them, to survive the subsequent heat shock. Now, coming to the heat shock factor 1, it's a transcription factor, as we all know. In the figure, it shows the survival of cephalic dopaminergic neurons with and without heat shock. From the first bar, as no heat shock has been provided, CAP neuron was not affected. But when heat shock was provided, the wild type neurons died out. But the preconditioned neurons, the neurons which are given the preconditioned treatment, still survived. This is all due to the expression of the heat shock proteins. Now, heat shock proteins and calcium channels. Small heat shock proteins mediate cytoprotection by preconditioning, as we have learned that this earlier. Golgi bodies are storehouse of calcium. So Golgi bodies, you all know, they store calcium. Heat shock protein 16.1 localizes to the Golgi, where it functions with calcium transporting ATPase to maintain a calcium homeostasis in medial Golgi. Now let's take a look at the panels on the screen. The heat shock proteins are fluorescently tagged, as well as the markers of medial and trans Golgi. Both the merged figures are different, though as from which we can see that the heat shock proteins are localized on the medial Golgi rather than the trans Golgi. And this is where all the activity of the calcium homeostasis is taking place. Cell death is mediated by high calcium levels. Disturbance of intracellular calcium levels is involved in necrotic cell death. To test this hypothesis, ITR1 gene that codes for the membrane-bound calcium channel is knocked down. Similarly, SCA1 gene that codes for calcium ATPase channel is also knocked down. Also, treatment with dendridine, a chemical, it inhibits the ryanodine receptor. The ryanodine receptor is also a calcium channel receptor, sorry, a receptor for calcium. You know. So from the figure, we can see that after the knockdown of these genes involved in calcium trafficking, apart from the control, all the untreated cells in the other criteria are high in number solely because of calcium homeostasis, either by the gene means either by the gene knockdown or chemical treatment. So discussion. HSP 16.1 is a general protector against multiple necrotic insults. What is the molecular basis of 16.1 protective function in the Golgi? That's the question. That's an interesting question. The answer to this would be this is because they act as molecular chaperones, efficiently binding denatured proteins and preventing protein aggregation. The calcium level is an important marker of the cells about to undergo necrosis, and malignant hyperthermia has been linked to mutations that cause calcium leakage in the cytoplasm. So guys, if you like this video, give it a thumbs up, and if you are involved in learning this kind of stuff, subscribe BioKit. Thanks.